This module will serve as a brief overview of the stages of the cell cycle, as well as mitosis and cytokinesis. Mitosis referring to division of nuclei within cells, and cytokinesis referring to cell division itself. When you think about a cell, such as we see on the left here, that is going to divide, obviously lots of things are taking place within that cell before and during cell division. However, one of the key features is that all of the chromosomes have to replicate. So on the left, if this nucleus has a complete set of chromosomes, those chromosomes have to duplicate themselves so that we can form two new cells, each of which has a complete set of chromosomes, and each of which is genetically identical to each other, as well as to the cell that we start with. So what we're looking at here is a diagram illustrating the major phases of the cell cycle. So imagine a single cell that again is destined to divide. This single cell will exist for a period of time doing whatever it is that that cell is supposed to do. The first period of time we identify is we refer to it as the first gap, or G1. All this refers to is the period of time before chromosomes begin to replicate. The time that this extends through can vary amongst different species, but again, the G1 phase of the cell cycle is just the time before chromosomes begin to replicate. At some point, DNA replication or DNA synthesis has to take place. We refer to this as the S phase of the cell cycle. So during this time, all the pieces of DNA, which of course are double-stranded, the two strands separate, complementary bases are added to each existing strand, and that is essentially what happens during chromosome replication or DNA synthesis. Obviously it's complex, lots of enzymes involved, but we won't dwell on those at this point. At some point, chromosome replication is complete, so the S phase of the cell cycle is complete. Then we enter a second period of time, the second gap or G2 phase of the cell cycle. Again, the length of this time can vary, but then the cell is essentially ready to divide, so that at some point mitosis can take place. Mitosis, division of the nucleus, and cytokinesis, division of the entire cell itself. So let's now turn our attention to some basic concepts that unfortunately can cause confusion at first sight, but hopefully we will discuss these and clarify some of these basic concepts at this point. What we're looking at toward the lower right is a schematic of a single cell. If you look at the chromosomes within the cell, notice we have a total of 6 chromosomes. Notice also that we essentially have 2 of each chromosome. So these 2 here, these 2 S-shaped ones, and these 2 V-shaped ones toward the right. So again, 2 of each chromosomes. What we're looking at here is a diploid cell. So most of the cells in your body are diploid. In other words, 2 of each chromosome. If this were a human cell, such as one from your body, we would be looking at a total of 46 chromosomes, or we might say 23 pairs of chromosomes. If this cell only had 1 of each chromosome, much like we find in sperm and eggs in humans, we would say that cell is haploid. Again, just having 1 of each chromosome. Also, if we take a look at any one of these pairs of chromosomes, we notice, for example, if we look on the far left here, these 2 straight chromosomes, we notice that they both have the same genes. They have the gene for hair color. However, notice that one codes for red hair, while the other chromosome codes for blonde hair. So we say they both have the same genes for hair color, but they have different versions of that gene, right? Depending upon the inheritance from mom and dad. So we can describe this pair of chromosomes as being homologous. So homologous chromosomes are those chromosomes that essentially contain the same genes. Again, that doesn't mean they're genetically identical, but again, it just means that they contain the same genes. They might have different versions of those genes, which we refer to as alleles. So alleles just refers to different versions of a gene. Also notice, give me a second here, I'll get my pen going. Also notice, if we take a look at a single chromosome, so I'm going to circle a chromosome in red. So that is a single chromosome. Notice it consists of 2 pieces of DNA. Again, this causes confusion at times, but that single chromosome consists of 2 chromatids. Each chromatid is genetically identical to each other. within a single chromosome. Why are there two chromatids there? Well, again, this is the condition after DNA synthesis takes place. If this were a cell in the G1 phase of the cell cycle, this chromosome right here, as well as all other chromosomes, would essentially exist as just a single piece of DNA, not two like we see here. So now let's briefly discuss some of the events that take place during the cell cycle. So I might have forgotten to mention it earlier, but we identified with the stages of the cell cycle, we identified G1, S, and G2. Collectively, G1, S, and G2, those all comprise interphase. So interphase, again, consisting of G1, S, and G2. Again, the time at which this exists varies, but interphase essentially is the time before mitosis takes place. If you look at the cell on the right, you're looking at this is an actual cell, a single cell nucleus well defined within that cell. This is a cell in interphase. Notice the DNA within the nucleus is there, it's present, but you don't see the DNA as very distinct chromosomes. It's not until a little bit later that distinct chromosomes actually become visible under the microscope. And so again, just to illustrate, if you look at the lower left, this figure represents a diploid cell, right, because there are two of each chromosome. Initially, on the far left, this is G1 phase. After DNA synthesis is complete, notice the cell on the right, still a diploid cell, two of each chromosome, and still just 4 total chromosomes. But again, at this point, each chromosome consists of 2 chromatids. That cell on the right is now ready to enter mitosis. So mitosis, we'll just identify 4 major phases of mitosis. And again, mitosis refers to division of the nucleus, typically is followed by cytokinesis or cell division. The 4 stages of mitosis we'll identify are prophase, metaphase, anaphase, and telophase. So now let's briefly outline what each of these stages of mitosis looks like. We will begin with prophase, which is essentially the second panel on this figure here. If we look at the schematic toward the top, a few things we'll point out about prophase. During prophase, the nuclear membrane begins to break down, the DNA condenses, and chromosomes become visible as distinct structures. Also, you'll notice in yellow, these structures here are centrioles. They start to move apart toward opposite ends of the cell, and these yellow lines that radiate between them, those are microtubules, part of the cytoskeleton. And so what happens, these microtubules begin to attach themselves to the chromosomes, and that pulls the chromosomes toward the middle of the cell. And so by metaphase, if we look on the far right here, by metaphase, all the chromosomes line up in the middle of the cell. Each chromatid has microtubules attached to it. Centrioles at this point, opposite ends of the cell. Nuclear membrane completely broken down by this point. Once all the chromatids have microtubules attached to them, then the chromatids separate and are pulled toward opposite ends of the cell. So after metaphase, we then have anaphase. Anaphase, shown here on the far left. Again, chromatids separate, pulled toward opposite ends of the cell. Finally, telophase, marked by 2 clusters of chromosomes. So one toward the top, one toward the bottom in this image here. Also notice that nuclear membranes begin to reform around those clusters of chromosomes, forming 2 distinct nuclei. And also, typically, but not always, typically telophase is followed by cytokinesis or cell division. So here on the far right, we essentially see the 2 cells dividing into 2. Just a few more comments about cytokinesis. If we take a look at cytokinesis in animal cells, the cell membrane essentially pinches in, thereby effectively splitting the 2 new cells apart. In plants, just a real quick comment, plants, a little different story because they have cell walls to deal with. In plants, once mitosis is complete, and we have 2 complete nuclei, then a new cell wall begins to form and eventually separates our 2 new cells that are produced as a result of mitosis and cytokinesis. So again, just to highlight, as a result of mitosis, the 2 new cells that are produced are genetically identical to each other and to the cell that we start with. This ends our brief overview of the cell cycle